In today's video, we'll be exploring two important clinical scenarios of how the body halts viral replication by targeting eukaryotic initiation factor 2 alpha and how to recognize a life-threatening lung infection in an immunocompromised patient. Herpes simplex virus produces a protein that activates a host enzyme capable of removing a phosphate group from the eukaryotic initiation factor 2 alpha subunit involved in initiating translation. This dephosphorylation restores protein synthesis, allowing the virus to replicate. Which of the following cellular mechanisms is responsible for triggering an antiviral response that halts protein synthesis by targeting eukaryotic initiation factor 2 alpha? Now, eukaryotic initiation factor 2 alpha plays a crucial role in regulating protein synthesis, particularly in response to cellular stress. And it's a key component of the translation initiation, and its phosphorylation status dictates whether protein synthesis is initiated or inhibited. The options given here are A. Interferon alpha, B. Interleukin 1, C. Interleukin 12, D. Transforming growth factor beta, E. Tumor necrosis factor alpha. The correct option here would be A. Interferon alpha. Before I explain why option A is correct, let's have a look at the steps of viral replication. In the first step, there is the attachment and the entry, in which the herpes simplex virus binds to specific receptors on the host cell surface using viral glycoproteins. It fuses with the host membrane and releases its capsid into the cytoplasm. In the second step, which is the genome delivery to the nucleus, the capsid travels along microtubules to the nuclear pore and injects viral DNA into the nucleus. The genome is circularized and begins the early phase of viral gene expression. In the third step, which is the viral replication and protein synthesis, the viral genome is replicated and proteins are synthesized in phases. And in the fourth step, which is the assembly and the release, new viral DNA is packaged into capsids, which gain their envelope by budding through host membranes, and mature virions are transported and released from the cell to infect new targets. Now, option A is correct because it is in the stage number 3 where double-stranded RNA may be detected, triggering interferon alpha responses. Interferon alpha promotes the synthesis of protein kinase R, which is activated by viral double-stranded RNA and phosphorylates eukaryotic initiation factor 2 alpha and therefore inhibits further protein synthesis. All the other options are incorrect because they do not promote the production of protein kinase R, which would phosphorylate eukaryotic initiation factor 2 alpha. For example, option B, interleukin 1, activates acute phase responses and is an endogenous pyrogen. Option C, interleukin 12, promotes type 1 T helper cell responses and activates natural killer cells. Option D, transforming growth factor beta, suppresses type 1 and type 2 T helper cell responses. And option E, tumor necrosis factor alpha, activates acute phase responses and is an endogenous pyrogen. A 62-year-old man with a history of chronic lymphocytic leukemia currently experiencing disease relapse presents with several weeks of coughing of blood and difficulty breathing. Vital signs include temperature of 38.6 degrees Celsius, blood pressure of 95/65, a heart rate of 110 beats per minute, and respiratory rate of 20 breaths per minute. On examination, he's thin, pale, and has decreased breath sounds on the left side. Chest imaging reveals a large lesion in the upper lobe of the left lung. Bronchial washings are cultured and the organism shown is identified. What is the most likely diagnosis? Now, this question gives several important clues. The patient is immunocompromised due to relapsed chronic lymphocytic leukemia, making him highly susceptible to opportunistic infections. The culture from bronchial washings is a key diagnostic step, helping narrow down the type of pathogen involved based on its structural and staining characteristics. The options given here are A. Aspergillosis, B. Coccidioidomycosis, C. Histoplasmosis, D. Nocardiosis, E. Tuberculosis. The correct option here would be A. Aspergillosis. And that is because this patient is immunosuppressed, placing him at risk for opportunistic infections like aspergillosis. The chest imaging shows a large mass consistent with an aspergilloma, and culture reveals acutely branching septate hyphae with conidial heads, which is characteristic of Aspergillus species. Let's have a look at why the other options are incorrect. Option B is incorrect because Coccidioides immitis is a dimorphic fungus and it appears in tissues as large spherules containing endospores and not septate hyphae. This patient's lung mass and culture showing acute angle branching septate hyphae with conidial heads are not consistent with Coccidioides. And option C is incorrect because Histoplasma capsulatum typically exists in tissue as yeast cells within macrophages and not as branching hyphae. While it can cause pulmonary nodules or disseminated disease in immunocompromised patients, it does not form fungus balls or produce the septate hyphae seen in Aspergillus. And option D is incorrect because Nocardia is a filamentous, weakly acid-fast, gram-positive bacterium and not a fungus. Though it can cause lung disease in immunocompromised hosts, it lacks septate hyphae and conidial heads, and it would typically present with branching rods visible on acid-fast staining and not with fungal structures. And option E is incorrect because Mycobacterium tuberculosis causes cavitary lesions, especially in immunocompromised patients. However, diagnosis relies on acid-fast staining showing red bacilli and culture grow buff-colored colonies, which are distinctly different from fungal hyphae with conidial heads.